The topic is regulation of gene expression. That means we are going to talk about the expression of a gene which is be regulated in both eukaryotes and prokaryotes. It is evident that when we talk about the cell, the genes which are present, they do not remain active throughout. They become active as and when they are required. Say, if a particular gene is required to produce a particular enzyme, then that gene becomes active. The other genes will remain inactive. That means the switching on and switching off the gene is under regulation. So there is a mechanism developed by the cell which stimulates particular genes to become active, and they switch off the other genes which is not required at that particular time. Because if all the genes become active, the whole thing is going to be very chaotic. And that mechanism is known as regulation of gene expression. So we have to remember that in the system, that when we talk about a cell, certain genes, depending upon their requirement to produce the enzyme, will become active. The other genes will remain inactive if they do not have to produce an enzyme. This has been studied both in case of eukaryotes as well as in case of prokaryotes. In case, first we'll take up eukaryotes, we'll not go in details about it. In eukaryotes, we know that the mRNA is monocistronic. That means it has one cistron or the gene which will encode for one polypeptide. So in this case, it has been found that the regulation of gene expression takes place at multiple levels. So we should remember that in this case it takes place at multiple levels. It can take place at the transcriptional level, where in this transcription level the primary transcript or primary transcript is formed, and in case of eukaryotes we call it as heterogeneous mRNA. And this is not the proper mRNA which will be used for further thing. So this has to undergo processing. So regulation of gene expression can also take place when processing of mRNA, that means heterogeneous mRNA, is taking place, and that is known as splicing. It can also take place when this heterogeneous mRNA is matured or has been processed. Then it is at that level this processed mRNA moves from the nucleus to the cytoplasm. So when it is moving from the nucleus to the cytoplasm, that time also regulation of gene expression can take place. And finally, it can also take place when translation is taking place. Translation means formation of protein, that is protein synthesis. So we will not go in details about eukaryotes, we will go to prokaryotes. Prokaryotes, gene expression has been studied in details, and it has been studied by Jacob and Monanti. They studied in 1961, and for this work, that means regulation of gene expression in prokaryotes, they got a Nobel Prize in 1964. And the prokaryote in which this regulation gene expression has been studied in details is Escherichia coli. One thing we have to remember here is that the regulation of gene expression can be affected by several factors. So one of the factors could be environmental factor, any of the environmental factors. The other one which regulates the gene expression would be the nutrients in the medium, and the most important, which we should remember, is the accessory protein. And this is the one which will regulate the gene expression in case of E. coli. In E. coli, we have to remember, or in prokaryotes, we have to remember that the mRNA is polycistronic. Many cistrons are there, and it will give rise to several proteins. And a very unique thing which is found in case of prokaryote is that the DNA segment comprises of, this is the DNA segment, it comprises of certain genes which are known as structural genes, and I am mentioning it as Z, Y and A. These are structural genes. Right upstream to it, we have an operator gene. O stands for operator gene. Then we have a P gene, which is known as promoter gene. And adjacent to it, we have something called I. This is regulator gene. And then we again have a P, or the promoter gene. This is the DNA segment. This whole DNA segment, along with the structural genes, regulator gene, promoter gene, operator gene, forms a structure which is now known as operon. So we have to remember that. What is an operon? Operon comprises, it's a small segment of DNA which comprises of structural genes, operator gene, before these structural genes, then we have adjacent to the operator gene, the promoter gene, and then adjacent to the promoter gene is the regulator gene. In case of E. coli, there are two types of operon which are studied, and those are lac operon and tryptophan operon. So in the next lecture, we will study details about lac operon and tryptophan operon.